Hello everyone, my name is Marianne. I'm a PhD student based in Brazil, and I'll be presenting some preliminary results of the UVC resistance in non-pigmented yeasts isolated from the Atacama Desert. Microorganisms have long been known for their importance not only for the ecological balance, but also for biotechnology processes. This is especially true for extremophiles, which can withstand extreme environmental conditions such as heat or pH, and thus are very useful for biotechnological processes. Besides thermophiles and alkaliphiles, the radio-resistant microorganisms have also been studied, not only because of the bioproducts that they offer, with special application in the pharmaceutical industry, but also because of the recent advent of techniques for synthetic biology, in which, broadly speaking, their molecular mechanisms of resistance could be applied to improve non-resistant microorganisms. Radio-resistant microorganisms can also be applied for studies on astrobiology, including the better understanding of how these could cope with the extreme parameters of extraterrestrial habitats, but also on primitive Earth. Ultraviolet radiation is primarily composed of three wavelength ranges: UVA, UVB, and UVC. Only the UVA and UVB ranges can reach the Earth's surface, and although being essential for all life on Earth, this can still cause direct and indirect deleterious effects, such as mutagenic lesions and increases in the levels of reactive oxygen species. Mars, on the other hand, is affected also by the ultraviolet wavelengths of the UVC range. UVC is even more deleterious, leading to higher rates of mutations compared to UVA or UVB. While we still cannot take these organisms to be studied in situ on Mars, the so-called analogous environments are still a very useful way of understanding how organisms can cope with such extreme conditions. In that regard, the Atacama Desert is an analogous environment with lots of similar conditions as what we would observe in Mars' surface. The high incidence of ultraviolet radiation is one of these parameters, and so the study of organisms coping with such extreme UV levels is a very good way of learning more about how life could survive on Mars. Most of the radio-resistant microorganisms retrieved from the soils of the Atacama Desert are pigmented bacteria, with melanin and carotenoids as the primarily found pigments. Non-pigmented microorganisms, on the other hand, still lack in studies and are only currently receiving the deserved attention. This is especially true for non-pigmented eukaryotes such as yeast isolates. Therefore, this study aimed to provide a first understanding of the resistance to ultraviolet radiation of non-pigmented extremophilic yeasts isolated from the Atacama Desert. Yeasts were isolated from the soils of the Atacama Desert back in 2012, and two of them were already characterized by Pushen and collaborators in 2015. So here we have a picture of two pigmented yeasts isolated during that campaign, Exophiala and Rhodosporidium, as well as the two non-pigmented yeasts, Naganisha frigimani and Otermaniella awatica. In their paper, Pushen and collaborators demonstrated with Raman spectrometry that neither melanin nor carotenoids could be identified in the two non-pigmented isolates. Later on, the team could also isolate 8 more of the non-pigmented yeasts from the same samples. These are yet to be identified and were used for the UVC experiments in this study. So the non-pigmented yeasts were cultivated in yeast media for 48 hours at 25 degrees Celsius and then replicated to solid media for the UVC exposure. 48 hours after the exposure, the colony-forming units were counted and compared to non-exposed control colonies. Because we don't have a radiometer in our lab, we used the growth of the previously characterized Naganisha frigimani as a pattern to estimate the UVC fluency being used. So as you can see in these graphs, Naganisha's growth is reduced in 1 log order after 30 seconds of exposure, which falls well into the 0.6 kilojoules per square meter observed on Pushen's experiment. Likewise, after 60 seconds of exposure, Naganisha's growth is reduced to 1/3 of the original growth, endorsing the estimates of a double fluency of 1.2 kilojoules per square meter. Thus, we can conclude that Naganisha's survival CFU numbers can be used as a reliable pattern for estimating the UVC fluence for our experiments. Then we exposed all yeast isolates in triplicates to the same UVC fluences of 0.6 and 1.2 kilojoules per square meter. And we can see from this graph that most of the isolates present a similar resistant pattern to what's observed with Naganisha. Some of the yeasts even presented a potentially higher resistance compared to Naganisha frangipani. This is especially true for the 13B isolate, with a reduction of only 1 log-fold even after exposure to the highest UVC dose. In conclusion, we can state that the growth pattern observed for Naganisha frangipani after UVC exposure can be used as an estimate for total UVC fluency. Yeast isolate 13B presented the highest UVC resistance and provides a powerful potential for further studies. All other yeast isolates presented overall resistance to UVC and endorsed the potential of desert, non-pigmented yeasts as radio-resistant organisms. These isolates shall be further explored for their potential as sources of bioproducts with importance for biotechnology, as well as for their potential as role models for astrobiology studies. Thank you very much.